All right, so welcome to today's Scientist to Go. My name is Aowen, and I am an educator at the Gulf of Maine Research Institute. Today, we are going to have a 25-minute or so presentation from Nathan, and then we're going to switch to the question and answer section. So as any questions come to your mind during the presentation, please go ahead and send those in the chat, and I will wait to save those until the question and answer section, and I'll read those off. And I guess without further ado, we'll get started. If you'd like to introduce yourself and start your slideshow.
Yeah, wonderful. Well, hello, everybody. I'm really excited to be able to share a little bit of my science with you all today. My name's Dr. Nathan Furey, and I'm an assistant professor in the Department of Biological Sciences at the University of New Hampshire. So I am really lucky in that I get to teach classes in things like ichthyology, which is the study of fish, as well as fisheries conservation and management. So how do we conserve populations that we harvest? And also stream ecology as well, which we'll touch on a tiny bit today as well. So with that, Eowyn, am I good to go ahead and start? Wonderful.
Yes.
So yeah, today we're going to be talking about fish movements and fish feeding, because this is a lot of the research that my lab does. But before I even dive into the science, I really wanted to start off with the idea that yes, I'm the one here talking to you about this science today, but really it's my team that does the science. And so I'm really lucky to work with a variety of undergraduate students, graduate students, postdoctoral scholars, technicians, state agencies, federal agencies, and other kind of academics really across the continent. So everything I'm showing you today is really the efforts of multiple people. But a little bit of my background and kind of how I got to be here, just here south in New Hampshire. I actually grew up in upstate New York. So I grew up in kind of the Finger Lakes region. I'm not sure if you can see my cursor or not, but that little red dot in New York. And so I was landlocked. But growing up, I just loved anytime I could be in the water, particularly going to any sort of beaches and seeing any sort of critters on the beach or in the water or in tide pools. So I decided I wanted to get degrees in marine biology. And so I first went to southern Maine, actually, in Biddeford, to the University of New England. And that's where I received degrees in marine biology and environmental science. And it was really there that I fell in love with research. The idea that I could come up with questions about how the natural world works, or, you know, what makes animals tick, or try to understand what they do, and do that for a career. So that's really when I decided, you know, I wanted to go to graduate school. I wanted to go on and get my PhD and become a professor myself. So then I moved from Maine all the way down to Texas. And I spent 3 years in Texas and received a master's in wildlife and fisheries science at Texas A&M University. However, then I wanted to continue my education and get that PhD. So we did another big move. And I ended up moving to Vancouver, British Columbia, where I did my PhD and postdoctoral work at the University of British Columbia studying salmon. And we're going to talk a little bit about salmon today and kind of why they're so cool as well. So that was kind of my path. And now I've been back in New Hampshire for about 5 years as a professor. So I've had a lot of fortune to kind of live in a lot of different places and in 2 countries, but very happy to be back in the Northeast United States. And hopefully this is obvious because this talk is about fish, but I love fish. I just think they're the coolest critters. They can have a lot of personality, and they just vary so much in their shapes, their sizes, their colors, what they eat, how they use habitat. We can find them in freshwater, saltwater, and everything in between. And we can find them in waters warm to cold. So this is just a sampling of the fish species that my lab currently does research on, and we'll be touching on these kind of throughout the talk. But in particular, I'm really interested in the movements of fish and kind of that behavior of fish. So kind of why do they move? Why go from one place to another? Where do they move to? And how can we actually know when we see a fish, like we release this fish from this bucket, how do we figure out where it goes from there? And so this gets to a big aspect of my research, which is kind of what we call movement ecology, and more specifically migration ecology. And so this term migration is really important in biology and ecology, but maybe it's not one that everyone has heard before. So the idea of a migration, and it's something that we don't see in just fish. We see it in all sorts of critters. So we see it in critters that walk or swim or fly, and we see it in critters like insects or butterflies that don't have backbones to our biggest and largest vertebrates, things like deer and elk and such. But simply a migration is when you have really large portions of a population or a large number of animals that are all of the same species going and moving at the same time and in the same direction and going to the same place. So we get these just large masses of movements of animals going from one habitat or ecosystem to another. And this just fascinates me. Like, why would animals do this? It costs so much energy and it's got to take so much effort to do that. And so what are kind of the consequences of this, or what helps dictate or determine the success of these migrating animals? So fish are no exceptions. Fish of all sorts of types also migrate. And we have really cool fish migrations, such as the salmon. The top left is an Atlantic salmon. The bottom right are sockeye salmon that are found on the west coast of North America. These fish actually migrate from the ocean all the way back into fresh water to complete their life cycle. So that's really neat, going hundreds of or even thousands of miles. In our backyard, we also have species like river herring that also migrate in from the ocean and come into our freshwater systems to spawn. And then we have species like the American eel that are also in our backyard that actually do the opposite. They actually migrate out to sea to spawn or to release eggs or to complete their life cycle. So these really large scale movements that can occur, literally over thousands of miles really fascinates me. And so if we want to know where a fish moves to, how quickly does it move, and whether it actually gets to its destination or not, we have some neat tools that we can use. And so my lab uses what's called telemetry, and specifically acoustic telemetry. And I know that's a lot of syllables there, but really all it means is that we're tagging a fish with an electronic tag, and then we can follow it around. So if we imagine this, this is a bull trout or a generic salmonid fish, we have this little tag that's in the photo there. We can implant that into the fish. And that tag actually makes a unique sound. It's not a sound that we can hear. It's at a frequency too high for our ears, but every minute or two, it will send out a sound signal. And so then we also need to put in the environment something to listen for that sound. And so we call these receivers or hydrophones or listening stations. And we can put these in the water. And their job is to just listen for those tags. And so if a tagged animal goes close enough to that receiver while it's sending out its signal, the receiver is going to pick that up and actually record it on its computer. So it'll say, okay, fish number 22 was detected on December 15th at 1:30 p.m. So we can know for each individual fish where it went and when, which I think is really, really cool. And so we can use this technology to track fish over pretty incredible distances. So this is a quick little video I'm going to try to play, and we keep our fingers crossed that it works, where we actually tracked hundreds of juvenile salmon. So kind of these fish on the right-hand side, we put these little electronic tags into them, and then we tracked their movements for over 600 miles through 3 river systems, and then about 150 miles along the coastline of British Columbia. So each of those little dots you're seeing run across your screen right now are little fish that have one of those tags and that we track over these distances. And so we can use this technology, and my lab does use this technology on a variety of fishes to try to answer a lot of questions about their migrations, or even just more generally their movements. What type of habitat do they use? What are their seasonal movements, etc. So that's on the West Coast, and I don't expect you to know your geography of Canada too well. So let's bring it a little bit more local here to the Gulf of Maine. So we've begun and been doing some tracking studies on a variety of species, kind of in the backyard of University of New Hampshire, and particularly the Great Bay Estuary. So an estuary is just simply where kind of the ocean and rivers or freshwater meets. And so we get kind of this semi-salty water, if you will, where the saltwater and freshwater meet. And we have a lot of important fishes that use this estuary for habitat to grow or habitat to spawn in. So an example of this is the rainbow smelt. And we have rainbow smelt here in New Hampshire, but along the coast of Maine, there are a lot of rainbow smelt populations too. And these are migratory fish that spend a lot of time in the summer and early fall out in the ocean in our coastal waters. And then in fall, they're going to work their way into estuaries and then up rivers. And then it's in late winter, early spring, they're going to spawn or release their eggs in those rivers. Unfortunately, their populations aren't doing very well, and we don't know a lot about their migrations and behavior. However, in Great Bay, at least historically, they were a really important fishery. People like catching rainbow smelt through the ice. And so you can go to downtown Exeter, New Hampshire on a cold enough year, and you'll see these little shacks on the water and these holes dug in the ice for people to go smelting. And it's because they're trying to catch these cute little rainbow smelt. And they tend to be served whole and fried. And I hear they're quite delicious, but I haven't eaten them before. And I just wanted to show this image because it shows that fish are in our backyard, even if we don't know it. So this picture was taken by Tim Briggs of New Hampshire Sea Grant by a drone. And here we're in downtown Exeter. And hopefully you can see in kind of the bottom center, a bunch of people on that little gravel bar. That is our lab as well as New Hampshire Fish and Game. And we are collecting rainbow smelt and measuring them and tagging them. And that's because that water that you're seeing there in downtown Exeter is their spawning habitat. So just in our backyard, off these old mill buildings and a really busy downtown, we have really important fish habitat. So this is important that we take care of our waterways. Even if we look in the water and we don't see fish, there probably are. So just like we did with those baby salmon, we can use these electronic transmitters to track the movements of rainbow smelt. So now in this video, you're seeing the movements of rainbow smelt after they've laid their eggs. And so it just simply allows us to follow these fish and see how do they use our estuary. And so we can see a lot of these fish use the estuary for a while, but now at the tail end of this video, we're watching them all exit. So these fish spawn in like April, and then they spend about 4 weeks or a month hanging out in the estuary. We don't know what they're doing, but we know they're there. And then it seemed like late April and early to mid-May, they all just took off. They all said, let's get back out to the ocean. We're done in this estuary. So this shows us how these tags can allow us to define things like, when do animals migrate? When do they use specific habitats, and what habitats are they using? And that's just another image of one of our tagged rainbow smelts. They really are beautiful, beautiful little fish. Oops. What's neat is that I've only shown you examples of little fish, but these tags can be used on big fish too. And so those receivers or listening stations that we put in the environment, we have those in Great Bay. We have those at Hampton Beach and Rye Beach. If anyone has ever gone to those beaches for like a summer vacation or something like that. And these receivers here, tags that other researchers put into bigger fish. And so this includes things like striped bass that are on the top left here, that migrate up and down the coast all the way from areas like the Carolinas and Chesapeake Bay, all the way up to New Hampshire and Maine. It includes the Atlantic sturgeon, which is a federally listed species as threatened, but these fish can get up to 10 feet long and several hundred pounds, as well as the white shark. So the white shark, if you've ever seen Shark Week or anything like that, these tags are also used on these animals to track their movements up and down our coast. And so there's been over 100 white sharks tagged by folks like Dr. Greg Scomal and Dr. Winton off of the Cape Cod of Massachusetts. Well, those fish come up to New Hampshire and Maine. Those big sharks come up there. They seem to just cruise on by New Hampshire. They aren't hanging out very often, but we always, a few sharks each year we detect on our receivers here near University of New Hampshire. And I've only talked about fish so far, but this technology or similar technologies can also be used for other marine animals, such as seabirds. So it's a little bit difficult to see, but in this image on the left-hand side, and I'm going to see if I can make a little pointer here, so hopefully you can see my screen a little bit better. You can see this little black trailing piece off of this tern's back. That is a GPS or satellite tag. So instead of sending out an acoustic noise, it sends a signal directly to satellites and allows us to follow around these birds. And so we're doing that to try to figure out, where do birds fish? Where do they like to catch fish that they can then come back and feed these to their chicks? Because the chicks, when they're born, they can't fly, they can't feed themselves. So the parents have to go out, catch fish from the ocean and bring it back to them. And that's what you're kind of seeing on this short little video on the right. It looks like a small little herring that this common tern parent brings back to its to chick to then gulp right up. And so my lab is doing research on the tern colonies at the Isles of Shoals, which is this beautiful set of little islands that are about 8 miles off the coast of New Hampshire and southern Maine. And they're home to New Hampshire's only seabird and tern colonies. So it's really important that we manage these populations and maintain them because seabirds are really important links in marine ecosystems here in the Gulf of Maine. And so one of my PhD students, Alia Caldwell, is leading this work. And now this video you're seeing are just the movements of these terns. And we can kind of see all the areas in blue are the places that we think those terns are feeding. And so we use some kind of complex statistical models to extract that out of the data. But if you just focus on where the blue dots are, we can see that these seabirds like to eat at river mouths along the coast. So again, these areas where saltwater and freshwater meet is where a lot of juvenile fish like to hang out. And that's a really good place if you're a seabird to go fishing. Alia is also doing her own fishing and seeing what types of fish can be found in these areas that are showing up as blue and relating that back to what the seabirds are bringing back to their nests so that we can start to identify when are they bringing back high quality prey items versus low quality prey items. Certain fish have a lot of energy and nutrients in them, such as herring, while other things don't have quite as much nutrients. Things like little bugs are really small and don't have as much nutrients and energy that the seabirds need. So this also gets to the next idea in my lab's research, which is that first, animals are moving to eat. So those seabirds are moving around so they can catch fish, but then what an animal eats and how much they eat is also really important. Just like you and I, what we eat, we want to eat a variety of foods and nutrition and all of that. Same thing for animals in the environment, that they need high quality energy to be successful. And my lab tries to understand how climate change might impact this. Is climate change going to impact what animals or fishes can eat? And that's really important in the Gulf of Maine because the Gulf of Maine is warming really rapidly due to climate change. So right in our backyard, we have that capacity to try to understand how is this affecting where we find different fishes or other marine animals, and is it affecting what they're eating? And so Another one of my PhD students, Nathan Herman, is actually using a data set that has the diets, so the stomach contents of over 300,000 fish that have been collected over the past 50 years. And he is looking to see how the diets or what these different fishes in the Gulf of Maine eat has changed over the past 40 to 50 years. Because every fish is going to have its own diet. Just like I'm sure what I eat for dinner is a little bit different from what you eat for dinner, it's the same thing among these fish. So something like the spiny dogfish here really is happy eating anything that it can fit into its mouth. And it's up in the water column. It's swimming around. It's not just hiding on the bottom. The monkfish can also eat a lot of different things, but it's going to hang out at the bottom. So it's going to probably be, it's going to encounter other different food items. So it's going to eat something a bit different. Something like the scup is up in the water column like the spiny dogfish, but it's a little more picky of an eater. It doesn't like eating as many different things as the dogfish. And then we have something like the witch flounder, which is on the bottom like the monkfish, but it has a really little mouth compared to the size of the monkfish. So even though it might want to eat a lot of different things, it can't fit a lot of things into its mouth. Fish don't have hands to be able to rip food apart, so they generally need to swallow their prey whole. And what we're seeing is that these species, not only are their diets different, but how their diets are changing due to our warming waters is also different. So the species on the right, they seem to be doing all right. Things like spiny dogfish that eat a lot of different species, or the monkfish that can eat a lot of different species, they're being able to maintain their quality of diet. They are eating just as well as they did decades ago. The species on the left, however, we're seeing them have a harder time keeping their stomachs full, and they're eating less than they did 40 years ago. So climate change is going to impact each species differently. So that's kind of over long-term changes, over 40, 50 years. But right now, it's been cold this week, and I know we're supposed to get some snow tomorrow. We're experiencing seasonal change. I also want you to know that seasons change in the water too, and this impacts how fish move and how they feed. So we're actually going to take a little bit of a detour to a part of the globe that experiences some of the most extreme changes between its seasons. And that's the Arctic. So if we go really far north, we get above what's called the Arctic Circle, and they have seasons where not only is winter cold, it is completely dark, 24 hours a day. And then their summers are light, 24 hours a day. So it's very extreme seasonality here in the Arctic. So how might that affect fish? So a couple of students in my lab are really lucky because they got to spend their summers all the way up north at this star in Tremblay Sound, Nunavut, Canada. And it is an absolutely beautiful landscape. However, the amenities or the housing might not be what you would want. There's no Wi-Fi, there's no TVs, there's not even any bathrooms. You have to go to the bathroom in a bucket the entire time you're there. Okay, so this is the field camp. They live together with about 30 scientists that spent their time in this absolutely beautiful landscape in the Canadian high Arctic. And what's really neat about this system and other Arctic systems is, again, that seasonality, from going from really cold and complete dark to warmer, not warm, but warmer and complete light. And what this does is it causes ice to melt. So during the cold, dark winters, this system is completely covered in ice, as you're seeing now, and then that ice in the spring breaks off really, really quickly. That allows a bunch of things to happen in the food web. Primarily, there's a lot of little bugs that we call amphipods that live in that ice. And so when that ice melts in the spring, all those amphipods get dropped into the water. And it's kind of like a feeding frenzy for all the fishes and other animals that live in that system. So when that spring happens, it's been cold, dark, and not a lot of food available to the fish. and then it gets warmer, and now there's food available, and the whole ecosystem responds. So this causes a lot of animals to migrate into the system. So this system is known by the local Inuit people because over 1,100 narwhal, so over 1,000 narwhal will come into this narrow sound each summer to feed. And that's because they're feeding on the fish that feed on the amphipods. Another really cool critter that comes into the system is the Greenland shark, which is a very large cold water fish that we think can live for hundreds of years. And I'm happy to talk more about that in the Q&A. However, my lab looked at 2 other fishes that use this system, and we wanted to know, did they feed on those little bugs that come out of the ice? And the answer is yes, they do, and quite a bit. So on the left here is kind of a normal sculpin in shape of a sculpin. This is what a sculpin looks like after it's eaten way too much. So its belly is completely full with those little bugs that came out of the ice. And I'm sorry, I hope this next picture isn't too disgusting, but this shows a stomach that's been cut open from an Arctic char, and all those little orange squiggly things are those little bugs or amphipods that come out of the ice. So these fish indeed are waiting for that ice to melt and drop down. I'm going to jump ahead a little bit because I know I'm running close on time here. But in short, we're able to track the movements of these char too and see that it's related to these ice dynamics. So these char want to be ready to eat when the ice comes off. And the sculpin, even though they're really little, they also move around quite a bit. I just want to end that we also do a little bit of work in freshwater. So hopefully in this image, you can see this adorable little brook trout here in the bottom right. But we want to know what type of habitat is valuable to brook trout. So streams, even though they're really small, they can be really important habitat for our fish. And so we have a lot of undergraduates in our lab, like Seal here, that go out and collect fish food in the streams. And that includes lots of little bugs. If you go into a stream in the spring or summer or fall and overturn a rock, I'm sure you'd find lots of little bugs underneath it. Those are all really good brook trout food. And then we can actually go out and we can talk more about this later too. We can go out and collect fish by using a technique called electro fishing. You look like a Ghostbuster and you actually shock the water a little bit to help you find the fish. And so that allows us to look at the size, the growth, and tag these little brook trout as well. So that kind of wraps up. I know that was a little bit of we bounced around topics and research projects, but I just wanted to give you an idea of all the different types of critters and systems we work with. But hopefully you learned that animals move and that they can move really long distances when they do this thing called migration, when they go all together at the same place at the same time. And that a big motivation in terms of why an animal moves, just like why do we drive to the grocery store or walk to the grocery store, is to eat. And also what an animal eats is important because that's how an animal gets energy and that's how they can grow. So with that, I know I'm kind of out of time. So I just want to thank you all for listening. And I just wanted to put my email there in case any of the classes wanted to reach out or ask any further questions beyond what we can talk about today. So with that, thank you all so much for listening. And I'm so excited to hear what you think and answer any questions you have.
Excellent. So we have quite a few questions. I don't know if you want to minimize so your picture will be bigger. But that might.
Yeah, I can go ahead and stop the share and I can pop back on if it makes sense too.
Perfect. So we've got lots of questions about some of the critters. So you just talked about the Greenland shark. So we've got a few good questions about that, including how long are they? How deep do they live and how long do they live?
Yeah, these are great questions. And the Greenland shark is just one of the coolest fish and one of the coolest sharks out there. And we don't know a lot about it yet, but I'm really lucky. One of my closest friends and colleagues, Dr. Nigel Hussey at the University of Windsor, is an expert in Greenland sharks. And so they can get quite large, 12, 15 feet long. These are big animals, and they are, even though they move really slowly whenever we see them in these really cold Arctic waters, they're really important predators. They have been found with all sorts of critters in their stomach, including polar bears. So they really are important parts of the ecosystem. They are known to travel quite deep. That we won't get into the details, but a lot of sharks are able to easily go from surface waters down in deep waters because they don't have what's called a swim bladder, like a lot of our other fishes have. So they can go down hundreds of feet deep as well. In fact, a few months ago, it was in the news. It was a very strange occurrence, but a Greenland shark was found somewhere tropical. I can't quite remember where, but it was because it was in the really deep water that was probably quite a bit colder, and it was pulled up from there. Now, in terms of how old they are, that is really neat too. Really old animals can be hard for us to age or to be able to estimate how old they are. But we have some kind of chemistry related techniques that can be used. And so there was a study that was published a few years ago that actually used some of the parts of the Greenland shark's eyes and used kind of what we call radiocarbon dating. So some of the same techniques that we use to kind of measure the age of like rocks and things like that. And they found that these Greenland sharks can live to be hundreds of years old. Now, there's not a lot of precision with that. We can't say that for sure that animal was 300 years old versus even 200 years old, but we know that it was likely hundreds of years old. And that implies that there were Greenland sharks swimming around at a similar time as to when, let's say, Christopher Columbus landed in North America. That some of those animals alive and swimming around today might have still been alive even when that happened. So we think this is one of the oldest vertebrates on the planet.
That is fascinating. I do remember when that study came out a few years ago about that Greenland shark. So cool. Speaking of kind of identifying things about sharks by looking at them, are you able to know the sex of a fish by looking at it?
Sharks, yes.
Oh.
Sharks, you can tell the sex of a shark by looking at it. A male shark will kind of have two appendages towards its anal fin, and those are called claspers. And so there will be a pair of those. And a female simply won't have those, and instead they have a part called the cloaca. Mm-hmm. For non-shark skates and rays, so those are all what we call cartilaginous fishes, and they're pretty closely related in terms of an evolutionary standpoint. We then have all of our other fishes, our other 35 to 40,000 species of bony fishes out there. They can be a lot harder to tell the sex apart. But a lot of these migratory fishes, for example, you can tell a little bit more because when it, particularly when it gets time to be spawning or to release those eggs, they can actually develop differences in their appearances. We call this secondary sexual characteristics. So for things like salmon, for example, the males get more of a hump-shaped back, and their jaws get kind of a hook shape to them. While the females, they stay more similar in their shape and coloration as well. So it really depends on the species if you're talking about the bony fishes. The cartilaginous fishes are very easy to tell. So again, those are the shark skates and rays.
That's cool. Thank you. What is your favorite species of fish?
Oh, goodness. This is a really tough one. I mean, in terms of the species I've worked with, I really like the bull trout because they are a fish of conservation concern. They really need cold, clean water to be successful. But they're also really flexible in how they live. So some bull trout are fine in a really tiny, small stream or river and live their whole lives there. Others can live their entire life in a big lake and get really, really big. And yet others still are migratory and will migrate all the way from a lake all the way out to the ocean. and come back as well. So those are really cool fish. Probably my coolest fish that I haven't had the pleasure of actually seeing, but I love teaching about it. And I'm actually am going to share my screen really quickly because I think I have this as a slide. Is the anglerfish. And so when we get into deep sea fishes, there are some really cool fish out there. And so this is the anglerfish, and it is perfectly suited to live in the deep sea. You can see all these long little appendages coming off of it and off of its fins. Those all act because in the deep sea, it's dark. There's no light. And so these act as little feelers. So that if something swims by, hopefully one of these little appendages will feel it and kind of tell that fish, ooh, something is around me. Is it something I can eat, or is it something that wants to eat me? They can also generate their own light. So this little appendage off of the front, hopefully you can see my cursor, that is called a lure. And there's actually bioluminescence in there. It can create its own light. So if we turn off all the lights, and that's how it is in the deep sea, we would actually see this little flashing light that would move back and forth, because it can also cast it back and forth, almost like a fishing lure. And that's how it gets shrimps and squids and other things it wants to eat to come to try to grab that lure. And then it's got, I don't know if you can see it. Nope. You can kind of see it here. They've got big, long, sharp teeth to grab any prey that get close enough to them. They also have a really cool mode of reproduction, but I'll stop there to try to get to more questions as well.
Cool. We've got a couple other like more specific things of do you like mako sharks and or clownfish?
Of course, I love all fish. So they are both really, really cool species, and for different reasons. So the mako shark, we think, is probably the fastest shark out there. You know, we think that they can burst at 50 plus miles per hour. And if you look at the shape of a mako shark, you can see why they're shaped like a bullet. They are perfectly what we call hydrodynamic. The water can just shed off of them and allows them to burst really, really quickly through the water. And so they're really well suited for their environment. They don't come very close to shore. They don't go near the bottom. They just like to swim in the open blue ocean for the most part. So they're perfectly suited for that. Clownfish are really cool because of their relationship with sea anemones and kind of their role within coral reefs. So sea anemones generally can sting other critters that would want to maybe mess with or try to eat a clownfish. However, the clownfish, with kind of their mucousy layer, as well as for a couple other reasons, are actually immune to those stinging cells from the sea anemone. So they can kind of live in the anemones and kind of make it home. And they're just also beautiful with those colorations as well. And I think I don't know a lot of detail about these species, but I do think there's some interesting social interactions that they have with other clownfishes as well.
Cool. Have you ever seen any squid, say maybe a strawberry squid?
I definitely have never seen a strawberry squid. I have seen a couple different types of squid. When I was doing my master's work at Texas A&M, we would do these week-long surveys off in the Gulf of Mexico, where we're just on a fishing vessel for a whole week. You're so far offshore, you can't see land at all. And we were actually out trying to catch baby fish. So we used really fine mesh nets to catch baby tunas and marlin and swordfish and things like that. But if you'd go out at night and turn on the lights, you could see squid hanging around near the surface of the water, which is just so cool. And we have neat squid species here in the Gulf of Maine, including the short fin squid and the long fin squid, that are really important parts of our marine food web and ecosystem here too. But no, I haven't seen a strawberry squid, which would be really cool.
I think so too. What is the largest species you've ever worked with personally?
Yeah. So I would say they weren't necessarily directly working with them, but I've handled them in a research capacity. So even during my undergraduate research, I worked on surveys to just try to understand what fish lived in our local waters in the Saco River in southern Maine. And we would catch Atlantic sturgeon in those surveys. Now we were catching smaller ones, so they were probably in that 4 to 5 foot range at most. But I've also gone out with commercial trawling vessels as well to do a little bit of research, and occasionally they would catch very large Atlantic sturgeon up to 8 to 10 feet long as well. And then I have had the pleasure of once helping tag a white shark. It was actually as part of an interview. It was with a lab out of California, and it was just part of the interview. They got a call that a juvenile white shark, so I think it was only 4 to 5 feet long, had got caught in some fisher's gear, and they wanted to know if I would go out and help tag it. And I said, of course I would. And so that was an incredible experience to work with that juvenile white shark and get it tagged and released safely. So those are probably the sturgeon and the white shark are probably the biggest fish that I've worked with directly.
Wow, those both sound like really cool experiences.
Yeah, and that's a picture of sturgeon in case you don't know what sturgeon look like as well. I guess
Sturgeon
I had one earlier.
Very neat fish.
They are.
So we have now reached kind of the end and my final 2 questions. So the first one is, what is your favorite part of your job?
Yeah, and so I think there's kind of 2 things that I want to touch on this. I mean, one is just getting to work with so many people who are as enthusiastic about fish and marine biology as I am. You know, growing up, I was a nerd, and, you know, I didn't necessarily have a lot of friends that thought fish or marine biology was really cool, especially in a landlocked part of New York. And so to kind of learn growing up that, when you graduate and you go on and whatever's next in your life, you're going to find people that are as interested in the things you're interested in, which is really exciting. 2, I love that every day is different. My job is never boring. Today I was making maps to try to understand where our fish went, and I was doing a lot of computer programming. Tomorrow I might be in the lab dissecting fish. And so, and then when we get a little warmer out, I'm going to be outside and actually handling fish and catching fish and all that. So I love that I have such a variety to what I do from day in and day out. I just never get bored.
That sounds pretty great. All right, so the very last question is what are you most excited about in your field?
Yeah, and so I've got a 2-part answer to this too. One is that we are getting more and more folks from different backgrounds studying fish and studying marine biology, where it doesn't matter where you're from, what you look like, you're even really what you're good at. As long as you're passionate about it and you treat people with kindness and you work hard, you have a place in marine biology. And I want you, if you're interested in studying fish, I want you to study fish too. And then on the other side, I think just the technologies are improving so rapidly and so quickly. This is everything from computing power and being able to deal with larger masses of data more quickly, but then even specifically with telemetry and these tags. All I showed you today is that we could follow an animal around, but we have tags now that can give us a lot of other information, like not only is the animal there, but what depth is it at? What is the temperature of the water? How quickly is the fish moving? Did the animal get eaten by something? We actually have tags now that can sense if an animal is eaten by something else. So the technology is just really rapidly improving. And I can't imagine what I'm going to be presenting, 10 to 20 years from now as the technology improves.
That sounds pretty cool. I'm definitely looking forward to that as well. Okay, I'm going to stop the recording.